Hi everyone! This video is all about introducing you to the neuromuscular junction. As you know, in order for skeletal muscle to contract, neuronal excitation of the muscle fibers must occur. Skeletal muscle fibers are innervated by large myelinated nerve fibers that originate from the motor neurons of the anterior horn cells of the spinal cord, also known as alpha motor neurons. As you can see here, each motor axon splits into a number of branches and comes into contact with the surface of individual muscle fibers. Although the nerve invaginates into the muscle fiber, it lies outside the muscle fiber plasma membrane. This structure is called the motor end plate. Each nerve ending forms a junction with the surface of the muscle fiber, and this together is known as the neuromuscular junction. So, clearly enough, you can see that there are two separate surfaces with a small space in between. Here, the neural membrane is at the top and the muscle membrane is shown at the bottom. The small space in between these two terminal endings is known as the synaptic cleft. It is 20 to 30 nanometers wide and contains extracellular fluid along with large quantities of the enzyme acetylcholinesterase. Let's look closely at the neuronal membrane. It contains mitochondria, synaptic vesicles, and voltage-gated calcium channels. Mitochondria are present in abundance, and they are responsible for the synthesis of ATP. This ATP is used as an energy source for the production of the excitatory neurotransmitter acetylcholine. Now, imagine this. There are approximately 300,000 synaptic vesicles in the neural membrane. They are synthesized high up in the soma, and then they travel down to the terminal ending. Not so coincidentally, acetylcholine is synthesized in the cytoplasm of terminal endings and is then absorbed into these vesicles. Now, as you can see, we have acetylcholine vesicles ready in the neuronal membrane. Now, understand this. The surface of the neuronal membrane contains gated channels for the exchange and movement of molecules. In this particular case, the membrane contains voltage-gated calcium channels that open in response to voltage or a change in membrane potential. When exposed to action potential, there is an enhanced permeability of the membrane to calcium ions, meaning calcium is now free to enter neuronal space. Now, let's look at the other end, the muscle membrane. It contains subneural clefts. These are basically numerous small folds that increase the surface area for the action of synaptic transmitter. I have a quick question for you. Where else in the body do we have similar folds that aim to increase the surface area? Think away, and I'll get back to this question at the end of the video. Coming back to the muscle membrane, it also contains acetylcholine-gated ion channels, also known as nicotinic acetylcholine receptors. As you can see, these receptors are located at the mouth or at the top of the subneural clefts. Now that you're familiar with the basics, let's quickly run through the chronology of events that essentially explain how we control our skeletal muscles. This is known as neuromuscular transmission. It might all sound complicated at first, but once you understand the flow of events, you will understand that it is very simple. Now, when an action potential is generated and reaches the terminal nerve ending, the voltage-gated calcium channels open and there is an influx of calcium ions. In response to this, acetylcholine is released from the vesicles. Remember, the amount of acetylcholine released will be proportional to the amount of calcium entering the neuronal membrane. Calcium ions exert an attractive force, so the vesicles are drawn towards the surface of the neural membrane. These vesicles then fuse with the neural membrane and empty their acetylcholine into the synaptic space by the process of exocytosis. 125 acetylcholine vesicles are released from the terminal at a time. The released acetylcholine then travels into the synaptic space and binds with the acetylcholine receptors on the muscle membrane. When acetylcholine attaches to the receptor, a conformational change occurs that causes the channel to open. This channel allows positive ions like sodium, potassium, and calcium to pass through it. Due to a high concentration and electrical gradient, sodium has most affinity. Now that the sodium has passed through the opening and is carrying a positive charge, it causes the membrane potential to change, in this case to increase slightly or become less negative. This creates a local positive potential change known as end plate potential, or EPP. When the end plate potential crosses the threshold or firing level AP, that is action potential, the action potential spreads along the muscle membrane, and this is what causes the muscle contraction. You must be thinking, what next? Well, for continuous functioning of the neuromuscular junction, vesicles need to be reformed again. So what happens to the acetylcholine? The effect of acetylcholine is rapidly abolished by the activity of the enzyme acetylcholinesterase, where acetylcholine is hydrolyzed to choline and acetic acid. So after contraction, about 1/2 of the choline is recaptured by the presynaptic nerve terminal and recycled to make more acetylcholine. Some of it diffuses out of the cleft. However, most of it is destroyed in the synapse itself by the enzyme acetylcholinesterase. However, within a few seconds only, acetylcholine is transported back into the vesicles and is ready to release for a new cycle of muscle contraction. That's it for the neuromuscular junction, but before we finish, let me answer the question. The inner wall of the small intestine is covered by numerous folds of mucous membrane called plica circularis. The surface of these folds contains tiny projections called villi and microvilli, which further increase the total surface area for absorption of nutrients. I sincerely hope this video helped you. Thank you, and see you in the next video.